On. Yeah, I'm good to go. Hello, everybody, here and online. My talk is going to be different from what you heard up to now. These are the things that I'm going to be talking about. You know, you've heard some of them, but I'm going to be talking from the pharmacology, toxicology, non-clinical reviewer point about all those issues. So, the drug review process is a multidisciplinary stepwise process involving evaluation of animal and human safety and efficacy data. It is very important to have evidence of safety and efficacy for every drug. It's not really ethical to get a drug on the market that is not safe or is not efficacious. So the PharmTox reviewer or non-clinical reviewer assesses the safety data that is submitted by the sponsor. So the stages of drug development, as you heard, and probably you all know, we start from pre-IND. Everybody's talking about pre-IND, so that tells you how important is pre-IND. IND, which covers phase 1, phase 2, and phase 3 clinical trials, the NDA, and post-marketing. Now for pre-IND, again, I'm going to emphasize that, like everybody else, you should really take advantage of it. It's a consultation. The way I look at it, it's a free consultation for you guys. I mean, FDA is answering your questions for free. And usually, it's a good thing to come as early as possible in the development process. So when the sponsor knows the property of the drug, has a drug made or a product at that time, it's called, not a drug yet, and have some non-clinical studies done or not even done. They just have a plan on what they're going to do, and they have a plan on how they're going to use the drug for what. Then you can submit for a pre-IND meeting to the FDA, and you have to write a package for the meeting. And the most important thing is that you have to put questions. If you just send us the summary without questions, we don't know what to do with it. We cannot answer you. So make sure you have questions. Some sponsors have 3 questions, some have 20 questions. It's okay. And then this is the advantages of the pre-INDs. You avoid premature submission of the IND. We can discuss any issues you have. Maybe there is some toxicity you've seen that you don't know if that's going to be go or no go. We can discuss it. So there's a lot of advantages of the pre-IND. So make sure that you take advantage of that. Okay, now the INDs and NDAs, you also looked at that. Or heard about it up to now from others. The IND, Investigational New Drug Application, the way I look at it, it is a request to conduct a clinical trial. That's when, you know, the sponsor has, again, has done some non-clinicals, done some chemistry, they have a product, and they have a clinical protocol, and they send us that information, and they're asking us, can we start the clinical trial or not? So, that's what PharmTox does. We look at all this data, we look at the non-clinical data, and we actually, we're really, I mean, I'm just going to say that, we're really the ones that would decide whether the clinical trial can go or not. As you heard from Judith, there's reasons for putting the IND on hold. Most of the time it is non-clinical. Sometimes it is chemistry, but because if it's not safe, it's from the non-clinical point. Then the NDA, the way I look at the NDA is a request to market a drug. By then, everything is done. All the clinical trials are done, the non-clinical done, the manufacturing, and the sponsor is submitting all this data and asking us, can we start to market the drug? So for us, for PharmTox, at the time, we're nearly done with reviewing all the non-clinical. Our job is to make sure that the information on the label is all correct. Because if you're familiar with our labels, there are information there comes from animal data, which is the carcinogenicity, mutagenicity, and reproductive effects. These are all from animal data. So that's our responsibility to make sure it's all correct. Now this is just gives you an idea of drug development and our time and the sponsor time. If you look at the white background is the sponsor time, and the blue is the FDA time. So the sponsor will start with preclinical research, will synthesize and purify a product, will do some animal data. There might be a pre-IND meeting here, a protocol, go to the IRBs, and then the IND is submitted here. This is our time, FDA, the 30 days that Judith talked about, and we will review the IND and decide if it is safe to proceed or not. And if it is safe to proceed, the sponsor will start their phase 1, phase 2, phase 3. There might be things happening here. Long-term studies will also continue. These are non-clinical. And during this time, even though this is all sponsor time, we have interaction with the sponsor all the time. Every protocol that they want to start or they write, it's sent to us. All the studies, non-clinical studies that are finished will be sent to us. So there's a lot, there are meetings here, a lot of meetings, so there's a lot of interactions. And then if something happens and something goes wrong, the sponsor can go on hold anytime here too. So when everything is done, then here the NDA is submitted, and this is our FDA time, again, to review the NDA. There might be an advisory committee here. If everything is okay, the drug will be approved. If not, we might go back to the sponsor to do some more work and comes back again, and so on. Now, just a little thing about biologics. Biologics therapeutics are composed of proteins, sugar, nucleic acid, or complex combination of all these things. And the biologics are isolated from a variety of natural sources. They're not chemicals. They're not made in the lab. They have to be from a human or animal or microorganism source. And they are, unlike chemical synthesized drug, most biologics are mixtures, and they tend to be heat sensitive and are susceptible to microbial contamination. So how does FDA regulate biologics? A lot of biologics are regulated in CDER, and those are the blood and blood products, gene and cell therapy, human tissue and cellular products used in transplantation, and vaccines. Now at CDER, what I have listed here under CDER used to be in CDER also, but in 2003, what we call the biologic therapeutics were moved from CDER to CDER. So what's listed under CDER, monoclonal antibodies, cytokine, etc., these are therapeutic. So any biologic that is used for as a therapeutic to treat an indication is now in CDER. Depending on that indication, it's in that division. So like I'm in the antiviral division, so we regulate both biologics and small molecules for antivirals, and so on for other divisions. So biologics versus small molecules, the differences between them, biologics are large and complex, small molecules, of course, are small, high immunogenicity to low immunogenicity. Biologics are highly targeted. They bind to a certain receptor. The small molecules are not. Biologic are species specific. When you're doing non-clinical studies, you have to have a relevant species. You cannot just use any species. And they're not metabolized, while drugs are metabolized. By biologics degrade to small amino acid, and they are excreted. And they have small volume of distribution, while small molecules have a large volume of distribution. And biologics are usually given IV, sub-Q, IM, while small molecules could be given the same way, and oral also. Most of them oral. You don't give biologics orally. Here's some example of the size, the comparison of size between some small molecules like acetaminophen compared to the coagulation factor. You could see how big they are. So now for our job as pharm-tox reviewers for safety assessment, we get pharmacology studies. So the pharmacology studies are divided into pharmacokinetic, which is what the body does to the drug, which deals with the ADME, absorption, distribution, metabolism, and excretion. Also, we have pharmacodynamics, and that's what the drug does to the body. So these studies, they're really, they're not for safety. If you look at M3, our guidance, they're not listed under safety. The sponsor, they do these studies. You all do them because you want to know the mechanism of action of the drug, the pharmacology of the drug, and you submit them to us. We review them, of course, because that helps us understand the toxicology of the drug when we know the pharmacology of the drug. These are the studies that has to be done for safety. And all this list of studies, they have to be GLP studies. The pharmacology studies don't have to be GLP. Those are the ones that need to be GLP. And they go from safety pharmacology, again, PK, ADME, general toxicology, local tolerance, genotoxicity, carcinogenicity, reproductive, and special studies if needed. Now, of course, for sponsors to know what to do, we have guidances, and I'm sure you're all familiar with guidances. And we have the ICH guidances, which comes from the ICH group. And the ICH started or established in 1990, and it was a group of pharma and the regulators in Europe, the US, and Japan. And now, of course, that's how it was when they started. But now there are other countries that joined the ICH, like Korea, Latin America, and I think lately China also joined them. So what they do is they meet, I think, 3 times a year, and they work on guidances. So there are guidances for safety, for quality, and efficacy. And you're familiar, like when you have an S, that's usually ICH S 5 or 6 and 7, this is for, safety, and quality is the Q, and efficacy, of course, is the E. And sometimes you have the M. The M is the multi-discipline. It covers everything. And here are where you find the guidances on the ICH guidance and the FDA guidance. And of course, we have ICH guidances, but then FDA has their own guidances too. And so does Europe and Japan and other countries. But if you follow the ICH guidance, your studies should be accepted in any country. This ICH guidances supersede any other guidance. And here's just a list of guidances. There's a lot of guidances outside, but I noticed none of them are non-clinical. I'm going to complain. But, but here's a list for you. And the studies I mentioned that need to be done, for example, safety pharmacology, which is to investigate potential undesirable pharmacokinetic effect on the physiology of the vital organs. And there's a core battery of studies done on cardiovascular system, respiratory system, and the CNS. And they're usually done in dogs or rats. PK, TK, same thing. These are the ADME studies. Usually they're done with 3 doses and a control, and you want to look at the metabolites, the metabolic pathways. The study should be similar to the route of administration of the human, how the drug is going to be used. And we need this data because later we're going to compare AUC and half-life with human AUC and half-life. So it's important to know these things. Then general toxicity, which is the most important studies for us, and this is single dose or multi-dose studies. And what we get from these studies is what is the target organ of toxicity? Is there a dose response for the toxic effect? We need to look at MTD. We need to go high. Sometimes the sponsors are afraid to go high to show toxicity. Actually, no, we want to see toxicity because just in case, we want you to go high and find the MTD. And also the no adverse effect level, which is used to calculate the first in-human dose. And also it's very important to know if the effect is reversible or not when we stop giving the drug. These tables are from our guidance, M3, and it tells you like the maximum duration of clinical trial. If the clinical trial is going to be 2 weeks, we have to have 2 species, a rodent and a non-rodent. Rodent could be a mouse or a rat, non-rodent either a monkey or a dog. So if it's up to 2 weeks, you need to have a duration of those animal studies 2 weeks and 2 weeks. And if you go 6 months, you have 6 months, and for the non-rodent, 9 months. The important thing is whatever is the duration of the clinical trial, your animal studies have to be equal or longer, not less. If it's less, that's a hold. It's not going anywhere. And now this is for marketing. We go a little bit longer. For 2 weeks, you need to do 1 month and 1 month, and so on. More than 3 months, which is considered chronic, the rodent is 6 months, and the non-rodent is 9 months. And that's the longest we go for a chronic in human. So genetic toxicity and carcinogenicity, mutagenicity and clastogenicity studies, they're simple studies done in vitro or in vivo, and these should be done early on because it will give us an idea if the drug is going to cause cancer or not. And remember, the clinical trials, the first clinical trial, most of the time is in healthy volunteers. So you don't want to give something to healthy volunteer that is genotoxic. So these studies are important, and it's important to have them early on. Carcinogenicity is done for drugs that are given chronically, more than 6 months, or given intermittently for life. For example, a drug for migraine, you need to do CAR studies, even though it's given maybe once a week or maybe once a month, but it's going to be given for a long, long time. And those studies are usually 2 studies in rats and mice. Repro studies, we have 3 different studies. The fertility of the adult animal, this is done early on. Embryo-fetal, and the fertility done in rats usually. Embryo-fetal development, these are the teratology study, and they're done in rabbits and rats. And the postnatal development of the offspring, and this is done in rats. And the first 2 are usually done during phase 2, and the last one is phase 3. And then special toxicology studies might be needed, depending. Maybe we want to know the mechanism of action of certain toxicity, immunology effects, phototoxicity. They don't have to be done, they're done only when needed. So when we have all this data as PharmTox reviewer, how do we look at it? Well, is an effect observed? Does it appear to be treatment related? Does it appear to be toxicological significant? Is it reversible? These are all the questions that we have to answer. Is it likely to be clinical relevant? And the most important question that our clinicians want to know, can the effect be monitored? Because when we see something and we tell our clinicians there's a necrosis in the heart, well, is that going to be monitored? So that's what they want to know. What can they monitor in the clinic? So the list I showed you of all these studies, they don't all have to be done by the IND. When you submit an IND, you need to do the safety pharmacology study, the acute toxicity, and the multi-dose toxicity. Most sponsors go straight to multi-dose. They don't need to do the acute, and the multi-dose need to be in 2 species. And then local tolerance if the drug is not given orally, if it's an IV or IM. And or sub-Q, and then the genetic toxicology, at least 2 of the studies of the genetic toxicology. So these are the studies that come with the IND. Don't have to do the carcinogenicity, the repro, or any of those other studies yet. These are tables for you. I'm not going to go through them all. Tells you what the study type, when it should be done, and for small molecules and for biologic. Do you need to do it for small molecules and biologic? For example, the second row, in vitro metabolic profile. Yeah, you need to do it early on for a small molecule, but you don't need to do it for a biologic because, like I said, they degrade and they go out. Same thing here, general toxicology. Before, of course, general toxicology is the multi-dose in animals before dose 1, 2, and 3, and you need to do 2 species for small molecules. For biologic, most of the time it's 1 species because that's the relevant species, and so on. So this is all information for you. And this one also is study type that you need to do for an oral administration or dermal or ocular. For example, if you look at the bottom, carcinogenicity, you need to do it for an oral if it's going to be given chronically. Dermal, yes, same thing. Ocular, may be able to waive if there's no systemic exposure, and so on. This is also information for you. Okay, now we've done these studies in mice and rats, so how are we going to go and calculate the first in human dose to this big guy from the little mouse or the rat we have? We have a guidance. It's the maximum recommended starting dose in human. This is an FDA guidance, not an ICH guidance. And it explains exactly what to do. You review and evaluate the animal data. You find a NOEL in the 2 species. Then you convert the NOEL to the human equivalent dose. And then you select the most sensitive species, and you add a safety factor of 10. So this safety factor of 10 depends. It's either, it doesn't have to be 10. It all depends on the toxicity. If it's a very toxic, if you have a toxicity like cardiotoxicity, you need way higher than 10. If you don't have, if it's only weight loss, you can live with less than 10. Okay, there was something here. Okay, calculating the human equivalent dose is, the way you calculate it is animal dose in milligram per kilogram multiplied by animal weight divided by human weight in kilogram to the power 0.33. I don't know if you guys know how to do this, but I don't. I don't know how to do this 0.33. So luckily, whoever wrote the guidance, the reviewers who wrote the guidance at FDA came up with this table. They actually used the same equation, which is down at the bottom, under the table. And they looked at, they used the equation, they used different weight range, body surface area for different animals, and they came up with numbers that we can use. So if you look at the last 2, here, the last 2, this is what you need. So to calculate that, you either, let's say take the mouse, which is the third one. So to to get the human equivalent dose from a NOEL in the mouse, you just divide by 12, or you multiply by 0.081, and so on. So it makes it really easy. So here's an example. If we had a NOEL in a rat study, was 10 milligram per kilogram per day. So the human equivalent is 10 times 0.162. This comes from the rats column. It gives us 1.62 milligram per kilogram. Or the way I like to do it is divide. I find it easier. So if you divide by 6, you get the same number, 1.6. Now this is milligram per kilogram. When we do experiments with animal, it's always milligram per kilogram. When you talk about humans, it's milligram. So we have to change that. So we use a body weight of 60 kilogram. So the human equivalent dose becomes 60 times 1.62 equals 97.2. Put a 10 factor, so we end up with 9.7 or 10. We do the same thing for the dog. The dog study gave us a NOEL of 5, same calculation, we got a 16. So now what do you recommend to the clinicians? Do you use a 10 or you use a 16 as a first dose in human? Come on. 10? Do I hear 10? Yeah, you always go with the safer. Go with the lower dose. And of course for us at FDA, it's the The sponsor does that, of course. When the sponsor submits the IND, they've done their calculation, they put safety factor, and it all depends on the NOEL they picked from the studies. So when we review the studies, we either accept that or we decide, no, we're not going to agree with this NOEL. We think there's a toxicity, the NOEL should be lower. So then the whole dose changes. So we can always go back to the sponsor and tell them to change the doses of the clinical study they're going to do. So now that we have done all this, our review, our decision becomes, were the non-clinical studies adequate in terms of dose and duration of the IND? Are there sufficient safety margins for identified toxicity? If yes, then we tell them they can, proceed with the study. If not, we put them on hold. Let's say we let them go. So, as I said before, the long-term studies will be needed. So the sponsor will be doing long-term studies and submitting them to us. They will finish their genetic toxicology studies. They're usually 3. If they started with 2, then they have 1 more to do. Their reproductive study, carcinogenicity study, all these things will be coming in. And then the NDA comes in when everything is done. Now, like I said, will be done by then because hopefully all these studies came through. Sometimes the carcinogenicity study might come with the NDA, so we have to review that. And our job as PharmTox will be working on the label. And when the sponsor submits the NDA, they always submit a label, a draft label, and then there's the negotiations of the label between the, on the label between the division and the sponsor. So we make sure our information, that's the carcinogenicity, mutagenicity, impairment of fertility, it's all correct. Pregnancy, we used to have pregnancy category before, now this is gone. We have to write a risk assessment in that area. And then we have another part on the label, which is animal pharmacology and or animal toxicology. This part, what we put there is if there is a if there's something that happened in the animals, but it didn't show up in the clinical trial, and we're really worried about it a little bit. So we put it in that area in case when the drug is on the market and thousands of people are, exposed, something showed up, so the physician will know what to expect. And then if everything is okay, we're okay with the NDA to be approved. We recommend approval. It's very rare that PharmTox will not approve an NDA because by then, if there is a problem, it will show early. Then it won't be lasting. It's usually chemistry that does that, or maybe a clinical trial. Maybe there's no efficacy in the phase 3. Okay, and then of course there's post-marketing. There might be some clinical or even non-clinical that we want the sponsor to do post-marketing, which is what you probably know as phase 4. Okay, now I have, how am I doing? I'm doing good for time. Okay, case studies. Now you guys are going to work. You're going to tell me, so pay attention, hold or no hold for these case studies. So we have a first-in-human and healthy volunteers to evaluate safety, tolerability, and PK in a single, ascending, and multiple doses. So the sponsor wants to use doses from 50 milligram to 200 milligram, single dose, and then multi-dose 3 times. Don't worry about that, just the single dose. So they've submitted safety pharmacology, genotoxicology, general toxicity studies were submitted. Everything was fine. So their general toxicity studies were 14-day rat, and these were the doses, and 14-day dog, and there's the doses. The CRO where they did these studies was not familiar to us. I've never heard of that CRO before. And we contacted OSIS, they are our inspection group at FDA, and asked them, and they haven't inspected that CRO. It was from outside the country. So the results we had was death at the low dose and the mid dose rat study poor overall conditions due to hydration. That was what the sponsor said. There was hydration, that's why the animals were not in good shape. And then we had vacuolation in the nervous system tissue, the brain, the optic nerve, and lesions in the lymphoid tissues, liver, kidney, lung, and spinal cord. So there was no NOEL in the rat study. So vacuolations in the nervous system, that's, I mean, how would you monitor that in a clinical trial? So the dog study was fine. There was a NOEL in it, and it was only based on increase in the CK levels and changes in ECG, so that was not a problem. So the sponsor suggested that the lesions are because of dehydration. That's all they said. Well, dehydration where? Is it the animals were dehydrated, or were the slides in the pathology dehydrated? We couldn't figure that out. So, hold or no hold? Oh, wow, good. You guys can go home. You know what you're doing. Okay, so, you know, when we put somebody on hold, we have to tell them what they need to do to get off hold. Sometimes that gets a bit difficult. So, and that's what we told them. The reason for hold is the vacuolation in the nervous system, and there was no NOEL, and that study was only 14-day study. And we said, even though you say it's dehydration, but really, I mean, it could be neurotoxicity. We don't know that. So, and here, the available non-clinical data is insufficient to support dosing in human at this time. So that was our conclusion. So what we told them to do, they need to repeat the 14-day study. They need to have the pathology evaluated by a peer review, which they didn't have that before, and to have a recovery group, and to try and go with lower doses, maybe they will get a NOEL. So they did that, and they actually were smart enough that they went to a different lab, and the study was fine, and there was no vacuolation, and we could, you know, let them go. And we still, you know, contacted our inspections, because when they did the last study that was okay, we asked our inspection about the lab. And they said, yes, this lab has been inspected and you can accept this study. And we also told them to make sure that to go and inspect the other lab, because we had other studies, like the safety toxicology study, the pharmacology study were done there. So we wanted to make sure that the other studies we can depend on them. Okay, so here were the safety factors from the new study. Finally, we had the NOEL and the rat, so it was okay. Okay, now this is a biologic, so I wanted to give you one of each. And this was for flu. And it was a monoclonal antibody and against the flu. And they wanted to do an open label dose escalation phase 1 study in healthy volunteers to evaluate safety and PK. And these are the doses 2, 5, 15, 30, and 50. And it's an IV infusion, single dose. So again, the non-clinical study, they had a mice study. They had 4 total IV doses on these days, 1, 2, 7, 14. And the dose levels was 5, 50, and 250. And there was a recovery for 4 weeks. So here we had mortality. 14 early death at the 5 milligram per kilogram dose. And also at the 50 milligram per kilogram dose. If you notice, this is the first dose, and the low dose, and the middle dose, and no early death at the high dose, the 250. So that was like really weird. I mean, you just, you know, you don't have a dose response or whatever. So, but you would think it's an immune effect. It's maybe, since it's a biologic, it could be an anaphylaxis or something like that. But of course, the sponsor does not explain that at all. So we thought it might be an anaphylaxis-based dose, and there were no follow-up work or anything explaining these deaths. They did a tissue cross-reactivity studies, and if you're not familiar with that, these are done for biologics, where you, especially in my division, this is for the flu. So a lot of the biologic we get in the division, the biologic binds to the virus. It doesn't bind anywhere in the body. There's no, so we really don't have a relevant species. So they do a tissue cross-reactivity study, which is a binding with different tissues, human or animal tissue, to see if there is any off-target binding or not. So they did that study, and their conclusion was that they did it with human and mouse, and they said that the sponsor said that the human and the mouse showed similar tissue binding, and no toxicity was seen in the mouse at the high dose, so then it's okay to use the high dose. So, so they used, that was their suggestion. So, hold on, hold. Okay, so they went on hold, and we told them that the death at the low and the mid dose, but not the high dose. Is it an anaphylaxis? I mean, wow, you need to explain it. And also the off-target binding. We had off-target binding in the GI, fallopian tubes, adrenal bladder, and kidney, which we're comfortable with. So then we told them to conduct another study with a different species. And so, well, maybe that one was with the mouse. Maybe there was something, with the mouse that maybe let them try a different species. And we told them if they see any anaphylaxis, then let them include measurements of immune complexes, immune globulin, serum tryptase, histamine, cytokine release. Let them show that this has really happened. And then we also told them to go longer than 14 days because, this is a monoclonal. Usually have a long half-life. The 14 days wasn't long enough. And I think they did all that, and the clinical trials are ongoing now. And I think that's all I have. Thank you. Have any?
There we go. Thank you very much. We're going to go ahead and take some questions now. We've got a microphone here, and I'm going to get another microphone set up over on the side of the room. So for those of you in the room, please line up at the microphones. We are going to enforce our rule of one question per person. And then if you have additional questions, you can go to the back of the line. Oh, we've already got somebody up there. Please, if you would, go ahead, step up nice and close to the microphone. Ask the question.
Long ago, it used to be that FDA wouldn't allow combination product studies under a single agent IND. Then that changed to allowing combinations under single agent INDs. Now with the new 1571, where those checkboxes only work when you check initial, does that mean that if you later in your development decide to, with a single agent, to then do combination products, you have to have a new IND?
Well, it depends. In my division, we're antivirals, so we have HIV and we have a lot of combination, drugs. So sometimes, they come with one and then they want to add another one, and sometimes we let them, like their first clinical trial, to be under that IND. And then when sometimes it gets too complicated, it cannot be followed, so we tell them to open another IND for the combination. So
it's a negotiable if you
it's a negotiable with the division. I would say that, yeah.
All right. And are you coming up? There you go. Step right up nice and close. Go ahead.
I had a quick question about if it's a targeted organ delivery.
I can't hear you.
If it's a targeted delivery,
there we go.
Like either in the eyes or in the ears, and in the cancer population, would you still require the two species? Would it have to be like a dog or a monkey? The second species?
For the eye, sometimes you can go with one species, yeah. And for cancer, also for the oncology, it depends. You're talking biologic or you're talking small molecules?
Small molecules.
Small molecules. It's all negotiable. I don't have that. It's a new ICH guidance S9, which is for oncology. Yeah, you're familiar with that?
No.
Yeah, I don't have it on my list. I should have updated it. That's a new guidance that would tell you exactly what to do for for oncology drug, and the same thing with anything for the eye. I would talk to the, to the group. Another, reason for a pre-IND, but sometimes you can get away with only one, if you have a reason.
Okay.
If there's no systemic exposure, sometimes it's only a local, and you can show it well in one species might work.
Thank you.
Okay, and I know we have an online question. For the online folks, remember that Q&A pod down at the bottom of the screen is where you put your questions in. Let's go ahead with the one online question we've got. And I'll be back over.
What is your recommendation for juvenile studies if the indication includes pediatric subjects? When is juvenile animal study not required?
I hate to say that, but it's case by case, and it all depends on the indication. And we have a pediatric, we have a guidance also for pediatric studies. If, again, in my division, if we know exactly what the toxicity of the drug is and we don't think it's going to be any different from, when given to pediatrics, then they don't need to do the study. Other divisions, I think because of their indication, they might insist on having a juvenile study. So that's another, just talk to the division if it's needed or not.
Okay, and we do have about 6 minutes left for questions, so if you have a question, please step up to one of the two microphones. Over there, please.
Hi. I see the nice breakdown you had as far as hypersensitivity allergic reactions.
The case study?
Yeah, as far as the case study. So being here in South San Francisco, we're all biotechs, and our big thing is immunogenicity. What kind of biomarkers do you consider appropriate or best practice when either confirming immunogenicity or what have you?
You have to take that into consideration. You have to show us if there's immunogenicity or not. We do understand that immunogenicity in the animals does not translate to humans. Most of the time, that doesn't mean you're going to have it in the clinical trials. The thing is, you want to make sure it's not affecting the PK of the biologic. That there's no toxicity because really there's no biologic there anymore because of the immunogenicity.
Right. That's fair. Thank you.
All right, the next online question, please.
Do we need to perform genotoxicity studies in animals, and they have in parentheses pre-IND, for oncology drugs as an example, small molecules?
Again, there's a guidance for oncology drugs, ICH S9, but a lot of oncology drugs are genotoxic, so they probably need to do something to know if it's genotoxic or not. They don't need to do carcinogenicity for those studies, definitely. But, it depends on the drug.
Okay, microphone number 2, please.
In a situation where you're trying to determine the max tolerable dose for a vaccine, say we chose 3 doses, we saw immunogenicity and no observed adverse events, but we still want to dose higher than our tox profile from our non-clinical studies. What's the best approach for making the argument to dose higher?
To making the argument, why would they not? I'm not familiar very much with vaccines. I don't do vaccines, but why would that be a problem of going higher in the animals?
So, from my experience, it's based upon at what dose level you did in your non-clinical would determine the tox, kind of that ceiling. And say we dosed at 1 × 10 to the 11th, and then if we wanted to do, say, go from a monovalent vaccine to a quadrivalent, and you want to do 1 × 10 × 4, you're now dosing at 4 times the amount that your tox data supports. Is there any reason for concern that we would get pushback from the agency for taking our dose times 4?
I can see why. You're still working with animals, right?
Yes.
Yes.
Yeah. Animal rule.
I don't see that, but then it's not my experience. I don't do vaccines, and this is, again, talk to the division.
Okay. Thank you.
Okay, let's take the next online question, please.
Can you accept clinical data generated in humans from another country for safety?
Human data, clinical. well, the IND, you have to open an IND in the U.S. and the sponsor have to do studies in the U.S. Our clinical trial, our clinicians look at all data that is done outside the U.S. So, yeah, I mean that would be looked at.
Okay. Any other questions in the room? Now is the time to come up to the microphone. All right, and online, then please help me thank Hanan.